What a lovely place you brought us to. I really feel privileged to be here. I'm so sorry I'm only staying in such a short time, but this is a beautiful place, and I'm sure that everyone's going to have a great time, and I'm quite jealous that I'll be leaving at 4 o'clock tomorrow morning. So I'm going to give my talk in 2 parts. The first part I'm going to talk about Alzheimer's disease and where we're up to in Alzheimer's disease, and where we are also in the treatment of Alzheimer's disease. And in the second part I'm going to talk about what Maria mentioned, how I think that genetics is now giving us a clue as to why in different disease processes different neuronal types are affected. So my talk is in 2 parts. So this is the pathology of Alzheimer's disease, the plaques, silver stained, and the tangles, also silver stained. The plaques are about 1/10 of a millimeter across, and they're made up largely of a peptide called the amyloid peptide. The tangles, as I'm sure you'll hear later in the week from Maria, are made up of a protein called tau. And besides these plaques and tangles, there's also cell loss, and of course that cell loss and synaptic loss underlies the dementia. And of course, when scientists saw this pathology, this complex pathology, they divided into groups as to those who thought plaques were important, those who thought tangles were important, and those who thought that neither were important, they were just the tombstones of dying cells. And our group decided to apply genetics to this problem. And what we did was we advertised in the Alzheimer's Society newsletter in the United Kingdom, and recruited families to our study. Families would write to us in our study and say that in their family they had multiple cases with disease. Which came first, the plaques, the tangles, or the cell loss? And we'd started to collect families. When we advertised in the Alzheimer's Society newsletter, we said if anybody has got more than 2 cases of Alzheimer's disease with an age of onset of under 60 in their family, could they write to us? And so, because molecular genetics was just starting at that time, and this family contacted us. This family has a grandfather with Alzheimer's disease, 2 sons with Alzheimer's disease, and those 2 sons married 2 sisters. That's actually not important, but it was the case. And then they had lots of children, 10 on this side of the family, and actually there were 6 on the far side of the family, but we just collected 3 of them. And all 6 of these affected individuals in black were alive at one time. And so we took blood samples from all of these families. And at that time, we just started to do genetic analysis. This was in the late 1980s, and we started to run genetic markers through the families. And what you're looking at here is the top half of chromosome 21, and you can see that all the affected individuals share all of that chromosomal region, but these 2 unaffected individuals, this person's got the top of the chromosome, this person's got the bottom, and that means that the only consistent finding is that this area of the chromosome is always inherited with disease, and in this part of the chromosome is the amyloid gene on chromosome 21. I'm showing you this because this is how positional cloning works. You don't need to know anything about the disease apart from its mode of inheritance. You just use the genetics to identify which chromosomal segment. The other point I'll make, though, is that when we did this work, there was a group of 6 of us, and this slide, which I'm showing now in 2 minutes, took 5 years of work. So what you're looking at in this slide is 30 person years of work. Nowadays, because of the whole the genome being sequenced, because we've got SNP arrays which allow us to test thousands of genetic markers all at the same time, because we know the human genome sequence completely, and because we have PCR, this was just, believe it or not, before PCR, we can do this much quicker, and now this would take a competent postdoc about a month to do. So in other words, the process of identifying genes is the same as it was 20 years ago, but the speed at which we can do it is 2 orders of magnitude faster. And this means actually, not only for Alzheimer's disease, but for every disease, that we can find any Mendelian gene easily now. All we need is the samples and the family structure, and it's a very straightforward process to find the gene. So the process is the same, but the speed is now very, very fast. Now, we found the mutations in the amyloid gene, the APP gene, and this led us to say, if you like, and this is the basis of the amyloid hypothesis, that looking at that complex pathology, amyloid is how that complex pathology starts, and everything else is downstream of amyloid. Now, in fact, we're not the first people to say that amyloid was important by any means. This was the first real statement about the amyloid hypothesis came from Glenner and Wong in this abstract in 1984. And what Glenner had done was he was the person who'd sequenced amyloid. In an earlier paper, he'd sequenced amyloid from an Alzheimer brain, and in this paper, he sequenced amyloid from the brain of someone with Down syndrome. People with Down syndrome always develop Alzheimer's disease when they're in their late 40s and 50s. He realized then in this paper it was the same sequence, and he wrote this: the amyloid protein from a case of Down syndrome was isolated and purified. Amino acid sequence analysis showed it to be homologous to that of the name has changed, the amyloid protein of Alzheimer's disease. This is the first chemical evidence of a relationship between Down syndrome and Alzheimer's disease. It suggests that Down syndrome may be a predictable model for Alzheimer's disease. Assuming the amyloid protein is a human gene, which it was, it also suggests that the genetic defect in Alzheimer's disease is localized on chromosome 21. And this remarkable abstract was of course completely accurate, but also makes the suggestion, the implicit suggestion, that cases of Alzheimer's disease can get Alzheimer's disease because they simply produce too much amyloid. And this really then is the amyloid hypothesis. I should say that I've circulated all of these slides, so all of you will have these slides. So this is the amyloid hypothesis, and I think we can be absolutely certain that the amyloid hypothesis is correct in Down syndrome. I think we can be reasonably certain it's correct in cases where you have amyloid mutations. I think we can also be reasonably certain that it's correct in cases with presenilin mutations, which I'll talk about in a second. But all of those are a trivial proportion of Alzheimer cases. They altogether are probably only 1% of Alzheimer's disease. Of course, the majority of cases of Alzheimer's disease occur in elderly individuals, and those elderly individuals do not have amyloid gene duplications, do not have amyloid mutations at all, and do not have presenilin mutations. And so it's a much more lively area of debate about whether the amyloid hypothesis is strictly correct in late onset Alzheimer's disease. And that's an important issue which I'll get to when I start to talk about treatments. So we found amyloid mutations, and now there's about 20 amyloid mutations. This French group, and we have now found 7 families in the UK, found amyloid gene duplications. This is shown here, and what you're looking at here is one of our cases, and you're looking at a gene chip, and I'm just showing you this just to show you how easy it is to analyze a gene chip. What you're looking at here is chromosome 21, all the polymorphisms on chromosome 21 from a single case, and you're scoring those polymorphisms AA, AB, BB here. And so this is homozygous A, AB, and this is homozygous B. But you can see over this area here, you have an extra chromosome where you can be AAB or ABB, and that is a duplication. That is a duplication. The amyloid gene is about here, in fact. So this just shows you how you can now instantly pick up these gene duplications in Alzheimer's disease and other gene duplications in other diseases. You can also see, if you look here, this is the density of hybridization, and you can see that the density of hybridization is greater over this area, showing you've got simply more DNA at this part of the chromosome. So this is how we now see gene duplications and gene deletions by an analogous method instantly on these chips. So this tells us that if you have the amyloid gene duplicated, you're going to get Alzheimer's disease at the age of about 55. There are interesting cases, a few of them, of people with Down syndrome who have got this part of the chromosome duplicated here. So they have a duplication down here and not a duplication here. And those individuals have Down syndrome, but they do not get Alzheimer's disease, except in the same way that the rest of the population can get Alzheimer's disease. So this very clearly shows, with both positive and negative data, that too much amyloid is going to give you Alzheimer's disease. Very interestingly, this mutation in amyloid was found by the Decode group in 2012, and this mutation, so here is the amyloid precursor protein here going through the membrane. This part from here to here is the amyloid in the plaques. It's about 40 amino acids. This mutation is here, and this mutation prevents amyloid being liberated from the amyloid precursor protein, and these individuals do not, well, actually they do not get Alzheimer's disease. They produce less amyloid, and they do not get Alzheimer's disease. The only case I know of personally is to do with a Finnish collaboration we have, and it's an individual who died at 102 years old with no amyloid deposition in their brain at all. So a very unusual case. So this mutation, this is, if you like, the opposite, a mutation which prevents amyloid production, prevents you getting Alzheimer's disease. And the implication of this is if you can stop amyloid production over a lifetime, you can stop Alzheimer's disease. That's the implication of this finding. Now, when we did this work, amyloid mutations, we'd collected, with my colleague Martin Rosser, we had collected 30 families with Alzheimer's disease, and the amyloid mutations occurred in 5 of those 30 families. So there was obviously other genes involved, and there was a race to find those other genes, and that race was won by Peter Hislop, who's now in Cambridge, and he found the presenilin gene. And what you're looking at here is in blue the structure of the presenilin protein. Here it goes through the membrane 9 times, and here, drawn as we now know it goes, is the amyloid precursor protein in the active site of presenilin. Because what we know is, although we didn't know it when it was cloned, is that presenilin is the enzyme Gammasecretase, which is responsible for liberating amyloid from the amyloid precursor protein. So this is the this is the, if you like, what you're looking at in this cartoon is the initial reaction which initiates Alzheimer's disease in these special cases. And all the residues in red are points that represent a single family where a mutation has been described. And in general, the mutations in presenilin tend to give an age of onset between about 35 and 45, and the mutations in the amyloid precursor protein tend to give Alzheimer's disease in the mid-50s. So each one of these red dots represents another family. Presenilin, just by the way, not only does it cleave the amyloid precursor protein, but it also cleaves many other type 1 membrane proteins. So many other proteins which have this structure with the N terminus going through the roof here and the C terminus going through the floor. This protein, gammasecretase, presenilin gammasecretase, is responsible for cleavage of many proteins in their transmembrane domain. And when we think of it as a drug target, that turns out to be an important complication, that this protein has many functions. I'll also say at this point that we really do not yet have a clear idea what the function of the amyloid precursor protein is. We have an idea that it's involved in synaptic contacts, but we really don't have a clear idea of what the precise function of the amyloid precursor protein is. And that is, I think, really a surprising deficit in our knowledge since we've known about the protein since 1987, so for nearly 30 years. The reason we don't really know what the protein does is because if you do a knockout in mice, you don't get a very clear phenotype. And the way that we usually try and work out what the function of a protein is by knocking it out in mice, and we don't have very clear answers from that type of experiment. So we only have a rather general idea about what the function of the amyloid precursor protein is. So this is a redrawing of that showing the amyloid precursor protein. Here is presenilin, and here in the transmembrane domain are 2 Ds aspartic acid, and in fact they are the active site of gammasecretase. These Ds here are the acid. They are responsible for the cleavage around about here. And we now have a very good idea, because of some great work by Bart de Strooper's group 2 years ago, a very good idea of how gammasecretase works. And I'll show you that in a couple of slides' time, but I'll just now talk about what the common features of all the Alzheimer-causing mutations are. Here you have the amyloid precursor protein, and it's cleaved by 2 major pathways. An alpha pathway involves cleavage in the middle of the amyloid part of the molecule. This is a combination of ADAM10 and ADAM17, followed by cleavage by presenilin gammasecretase. This is the beta pathway here. The enzyme base, beta secretase, cleaves here, and that yields amyloid. So these are the normal pathways of metabolism. The first mutation which was worked out was a mutation we found here at the N terminal, and that is simply a better substrate for this pathway. So more goes down this pathway. So again, these people simply make more amyloid, and that's why they get sick. And that's how this, and the Icelandic mutation that I showed you in the last slide is exactly the opposite of this. It pushes things down this pathway here. And then this is the mutation we first found in that English family. It's just outside the C terminal here, and it doesn't alter the flux through the pathway. What it does is it marginally alters the position of this final gammasecretase cleavage, so we produce more of a longer amyloid, and that longer amyloid is less soluble and therefore more prone to deposition. And presenilins in general have the same effect, but I'm going to show you more details about that in the next slide. And then there's a series of mutations here in the middle of the amyloid part of the molecule, and they are worse substrates for the alpha pathway, and so more by default goes down the beta pathway. So the mutations, the three different clusters of mutations, have different molecular effects, more through this pathway, longer formed, more prevalence, less through this pathway, but they all have the same outcome, and that is amyloid deposition is more likely. Now, we actually now have a very good idea. There's been a lot of work on Gammasecretase, particularly done by Bart de Stroeper's group, and I would really recommend this excellent paper in the EMBO journal from a couple of years ago. And what he has shown is, in fact, what his group have shown is, in fact, that the cleavage of APP is very complicated. There is an initial cleavage which can happen. So here is the N-terminal of the amyloid part of the molecule, and that's A-beta 40 there, and this is A-beta 42. These are the proteins you find in the brain. But what he has shown is you get initial cleavage at the amino acid 49, and then when you have cleavage at 49, it then subsequently cleaves again and again and again at each turn of the helix. So you get sequences with 46, 43, and then 40 amino acids. Or you can have the initial cleavage at 48, and when you get initial cleavage at 48, you then get cleavages at 45 and 42. And so you have the cleavage by presignaling gamma-secretase of APP is a complex event. Actually different mutations have subtly different effects, but all of them result either in producing more of A-beta 42 by pushing into this pathway, or by producing more of 43 by inhibiting these subsequent cleavages. And so the actual effects of presenilin mutations are complex, but the outcome is simple. You get more of a longer amyloid produced, and that longer amyloid is less soluble. And this is a beautiful paper. I really recommend you look at it. It's free online, so you can download it now if you're bored. So, this is the amyloid hypothesis then. It's clearly very solid, I think, in Downs and APP and presenilin mutation families. I think that if we're going to test drugs against the amyloid hypothesis, we should actually test them first in these families where we know, or in Down syndrome, where we know the hypothesis is operating. It's a clean form of disease, and these individuals clearly have initial amyloid, a problem initially in their amyloid metabolism. In fact, in the early trials, which occurred in the late 90s and early 2000s, these people were excluded from the trials because the trials were done in typical late-onset Alzheimer's disease. And I think that that was a mistake. And I also think it was very unfair because the woman from the family I showed you, who brought her family to us for analysis, said that she hoped that this would mean that her family would be involved in the first drug trials, and actually they were excluded from the first drug trials. So it was really unjust in my view that that happened, but it happened for reasons which are defensible. Clearly, later, other pathologies can become sustaining and self-sustaining and propagating. I would imagine that you will have lectures on that this session, but there's been excellent work on both tangle pathology, tau pathology, and synuclein pathology, showing that once the pathology, you can initiate these pathologies by injecting human tangles into mouse brain. I imagine that Maria will talk about that. Maybe if she wasn't going to now, she will. These pathologies become self-sustaining. So this is really, it's really important, I think, then, that if the other pathologies can become self-sustaining, we have to get at treatment early. And the other thing that has always worried me is why is it in late onset disease where we don't have amyloid mutations, why do those individuals get Alzheimer's disease? Is there a reason behind their amyloid deposition? And could that reason be something related to the function of the amyloid precursor protein? But I don't have an answer to that, but that's a concern, and there's been some very good work from the Washington University group, headed by Randy Bateman, showing that in late onset Alzheimer's disease, the problem is not amyloid overproduction, but the fact that clearance from the brain is much less efficient, much less effective. So there's a problem in clearance, which his group has shown. Now, now with this background, I often get asked to debate the amyloid hypothesis, and the reason is there's been 7 trials which have purported to be amyloid trials, and none of them have passed their primary endpoints. So none of them have worked. However, and we argue that in here, and again this is freely available online, of those 7, of those 6, only 2 were actually on target. In other words, 2 of the drugs were given at too low a dose to work. In other words, they were given at doses which have been ineffective in mice. One of them was never shown to be effective in any system. One of them failed for side effects unrelated to amyloid, and 2 of them were, if you like, proper trials and did not show efficacy, but did not pass their primary endpoint. So we've had 2 shots on goal, and neither has scored. That, of course, is a concern. We have, however, learnt quite a lot from these trials. The first is that, and perhaps the most important thing, is that we were let's go back up a bit. 2 of the trials showed evidence, not statistically robust enough, but showed evidence for efficacy in early individuals very early in the disease process. However, on analysis at autopsy and through amyloid scans, both trials showed that the misdiagnosis rate in very early individuals, in other words, the accuracy in diagnosing Alzheimer's disease in very mild individuals, was only 70 to 75%. In other words, in those trials, 30% of the individuals who were diagnosed as having mild Alzheimer's disease, in fact, did not have mild Alzheimer's disease, and either had something else like depression or frontotemporal dementia, but they did not have Alzheimer's disease. And so any signal in these in these mild cases would have been diluted by the misdiagnosis rate. And what this is going to mean going forward is that these trials are now being, 2 of these trials are being repeated, but with amyloid scans, which are quite expensive, to check the diagnosis in early individuals. So that's where these trials are up to. The other thing that we have learnt over the last, I would say over the last 15 years, is that the complexity of the relationship between the pathology of Alzheimer's disease and the clinical symptoms. And that complexity really first came again out of work from Down's syndrome, where David Mann showed about 30 years ago that amyloid pathology started in Down's individuals in their early 30s. And this, from a paper by Cliff Jack from the Mayo Clinic, shows the relationship that's now, well, it's with a lot of evidence between the pathologies of Alzheimer's disease and the clinical symptoms. And so this is the detection threshold here. So here, this is when you would make the diagnosis of someone with Alzheimer's disease here. This is without a timescale, because obviously with presenilin mutations, this would be somebody at 45. In the general population, this could be 75 years. Here we have the beginning of, if we did an MRI and did fluorodeoxyglucose PET, we would either see a bit of hippocampal atrophy or reduced cerebral blood flow, and this is about 5 years before the onset of clinical symptoms. If we did a CSF tap, took CSF, what we see in Alzheimer's disease is a raised CSF tau, and we would start to see that here, which is about 10 years before Alzheimer's disease. So that is saying that we're beginning to see abnormalities in the tau system about 10 years before Alzheimer's disease. And if we did either amyloid PET or we measured CSF A beta, we would start to see changes in amyloid metabolism 15 or 20 years before we started to see clinical symptoms. In fact, the CSF change that we would see is a reduction in CSF A beta, because it seems as if what is happening is that the amyloid which is being produced at this point, instead of being coming out through the CSF, is actually depositing on the incipient plaques. And so what you see here, about 20 to 25 years before the onset of clinical symptoms, is a gradual reduction in CSF A beta. At the same time, PET scanning can pick up the beginning of amyloid deposition. Now, this time course really poses an enormous number of practical problems and theoretical considerations. For a start, you know, one of the implicit things about the amyloid hypothesis is that amyloid is toxic. That's what you kind of think, that's the simple-minded idea you might have. And yet, you've got amyloid in your brain for 20 years before you have Alzheimer's, before it's clinically apparent. It's very difficult to think of a process. I mean, what we have to do is think of a process which is going to take that length of time to initiate serious problems. And clearly, an acute toxicity is not really likely. The other point, though, is that if we really want to stop Alzheimer's disease, if we really want to and have an amyloid attack, you know, it's better to be starting to deal with amyloid in this part of the curve here, rather than waiting here for when we have diagnosis. So we want to be starting to treat the disease at periods, well, actually, ideally, before you have clinical symptoms, and that has also many implications, both ethical and practical, in terms of identifying people. Two solutions are being tried immediately. Well, they're really variants of one solution, and that is to take people who are genetically predisposed to Alzheimer's disease and start treating them with, for example, beta secretase, or, well, it's beta secretase inhibitors long before they have disease. And so there are studies, there's the Diane study out of Washington University, and then there's the Alzheimer's disease prevention strategy from, which is dealing with a large family with a presenilin mutation from Columbia. So there are two studies going on where treatments are being initiated before disease onset. But obviously we have to think very carefully about whether we giving treatments which may have side effects. In fact, most treatments have side effects to individuals who are completely well when we're giving them the treatment. So that's where we're up to, I think, with the amyloid hypothesis. I'll now move on to the second part of my talk, which is to think about selective vulnerability. You know, we're living in it's a great time. This is the best time ever, I would say, to start to study neurodegenerative diseases as a cell biologist or as a protein chemist, or as a systems biologist, because, you know, in the old days we had one gene. You know, we had one gene well, we had the APP gene, and then we had the presenilins, we had 3 genes. Now, because of genome-wide association studies, and because we have so many different genes for any individual clinical syndrome, we are faced with a banquet of genetic risk loci. We have a huge number for any syndrome, a huge number of genetic risk loci. And this gives us a richness when we plan our experiments that we have never had before. This started to come out of the Alzheimer's disease genome-wide association studies. So what I've shown you is the identification of 3 genes for early onset disease: amyloid, the presenilin, and well, actually I only mentioned presenilin 1, but there's presenilin 2 as well. And they don't explain any really of late onset Alzheimer's disease. So everybody working on Alzheimer's disease, collecting late onset cases, started to pool their sample series in about 2005. And now we have, in Alzheimer's disease, collected this is the most recent analysis. There's another one just about to be reported, but not much different. We have collected 30,000 Alzheimer cases and 44,000 controls with typical late onset Alzheimer's disease, mainly from Western Europe and North America. North America. And what we have done is we have tested 500 SNPs from across the genome using the chips I showed you before. So we've tested 500,000 polymorphisms simultaneously for association with Alzheimer's disease. And the way this test works is extremely simple. What you're looking for is polymorphism which occurs more frequently in cases than controls, or the other way around. That's all you're looking for. But because you're testing 500,000 SNPs, you have to set your Bonferroni correction. Bonferroni was the cruelest Italian, let me just say that. So you have to test, you have to correct for 500,000 statistical tests. And that means to prove something to anybody's satisfaction, you need to get to a p-value of 10 to the minus 7. And what you're looking at here is the log of p-value, and this is the line at 10 to the minus 7. And here you have on this axis the chromosomes, 1, 2, 3, and so on. And then these, when a polymorphism exceeds that 10 to the minus 7, then we're allowed to declare it as a gene for disease. And this is the data from 40,000 cases and controls. I'll firstly mention ApoE. Here is ApoE. ApoE was identified by Alan Roses in 1994 as associated with Alzheimer's disease, and actually its association is so strong that it would actually go way, way, way through the roof. It's far stronger association than any of the others. But these are the other genes that we have now identified. And we have now identified somewhere between 30 and 40 other genes for Alzheimer's disease. So we've identified now 30 or 40. A list of genes, however, is rather boring because really what we're interested in is processes. So what do these genes have in common? And the answer is, and we could see this from the first 7, this is the first 7 which were identified. ApoE was identified before, as I mentioned, cholesterol metabolism. The next one was clustering. This is also involved in brain cholesterol metabolism. It's also involved in the innate immunity and the complement cascade. This was the next identified, then PCALM. This is involved in a process called endosomal vesicle recycling. ABCA7 was the next gene to be identified. This is involved in cholesterol metabolism. In fact, it's a homologue of ABCA1. Mutations in ABCA1 cause a cholesterol storage disease called Tangier's disease. So this is clearly a cholesterol metabolism gene. CR1 is involved again in innate complement receptor 1, innate immunity. BIN1 again in endosomal vesicle recycling, and then MS4AE, innate immunity. So you can see that although we've got these 7 genes, they all map to those 3 pathways. And the same is true now. Now we're up to 30 or 40, the same holds true. So what this is telling us is these are the processes which are underlying Alzheimer's disease. And actually, I don't know if I've got this slide on or not, but if you look at the genes which just fail in genome, just fail to reach the 10 to the minus 7, so these don't reach Bonferroni, we see a pile of genes here which map to the same pathway. So this enables us to identify more genes, even though they don't reach the Bonferroni correction. We have to be a little bit careful because it's difficult to cheat Bonferroni, but it gives us some indication that these might be real hits. So what this is telling us is that the genes for Alzheimer's disease map in 3 pathways. And I'm not going to talk about it tonight, but we, by exome sequencing, identified a gene called TREM2 as involved in Alzheimer's disease. And TREM2 is a gene involved in the innate immune system in microglial activation. And so the genes that are coming out of the sequencing results fit into those 3 pathways. And so this is telling us that Alzheimer's disease, to some extent, is a disorder in these 3 pathways. Exactly which members of these pathways and how these pathways all interrelate and how they relate to amyloid deposition, we don't precisely know yet, but it's telling us that there are pathways to disease, and these are those pathways. And actually, if anybody reported an Alzheimer gene which was not in one of these pathways, I would be very suspicious that they were not correct. We're doing exactly the same thing in Parkinson's disease. In Parkinson's disease, we're doing genome-wide association studies, and we've now found about 30 loci for Parkinson's disease. And again, these are mapping to pathways. And the pathways they're mapping to are the ones that we understand are lysosomal biology, the immune system, and mitophagy. So again, we're beginning to see pathways to disease, different pathways leading to Parkinson's disease. So we're seeing commonalities. Now, in Parkinson's disease, the pathology of Parkinson's disease is Lewy bodies. And Lewy bodies are made up, in fact this was Maria Spallanzini who showed this first, Lewy bodies are made up of a protein called alpha-synuclein. And we found alpha-synuclein mutations in some Parkinson's kindreds. And when we did the genome-wide association study, here you see the synuclein gene on chromosome 4. So normal variability in the synuclein gene contributes to the risk of getting Parkinson's disease. So a very clear relationship between the pathology of the disease, synuclein deposition, mutations in synuclein causing Parkinson's disease, and normal genetic variability predisposing to Parkinson's disease, with those of us who produce more synuclein being more at risk for getting disease. But again, the emphasis I want you to take away is that we're seeing pathways to disease. Now, dementia with Lewy bodies, however, dementia with Lewy bodies is very closely related to Parkinson's disease. The pathology of dementia with Lewy bodies is the amyloid of Alzheimer's disease, the amyloid of plaques of Alzheimer's disease, but instead of lots of tangles, you have Lewy bodies. So what we did in our center recently was to do a genome-wide association study of dementia with Lewy bodies. This is a common disease. It's about as common as Parkinson's disease. It's defined usually pathologically, so we took pathological samples, and we did a genome-wide association study for dementia with Lewy bodies. And what we found, and it's not as impressive as the others because the numbers are still smaller, we're still doing this, was again the synuclein peak on chromosome 4 here. This is also another peak at a gene called SCARB2, which is a lysosomal gene here. So we saw two peaks here. This is the ApoE peak because you've got the plaque deposition, which seems to be driven somehow by ApoE. But here you've got synuclein and this other gene, SCARB-P. When we looked closely at the synuclein peak and compared the synuclein peak in Parkinson's disease with the synuclein peak in dementia with Lewy bodies, this is what we saw. And this is blowing up that peak to a great extent. And in red, this is the association with Parkinson's disease. Here's the synuclein gene. Here's the 5', here's the 3' end of the synuclein gene. And these are the SNPs which are showing association with Parkinson's disease. In blue are the SNPs which are showing association with dementia with Lewy bodies. And you can see there is an association with synuclein, just as there is in PD, but the association is different. It's to a different part of the gene. So in Parkinson's disease, it's to the 3' end of the gene, and to dementia with Lewy bodies, it's at the 5' end of the gene. This is the other gene I mentioned briefly, and you see the same effect. Here's the Parkinson's disease association. Here it's at the 5' end of the gene, and here is the association with dementia with Lewy bodies. It's at the 3' end of the gene. So you were seeing in these two disorders, which have fundamentally the same pathology, we're seeing different parts of the gene showing associations. Now, when we look in these genes, we don't find amino acid changes. This is not driven by amino acid changes. What you're looking at here is some type of genetic variability in expression. And we can't distinguish exactly what is behind this yet, but there are two alternatives. The first alternative is that what you're seeing in red is what's driven in the nigra. In other words, you're seeing genetic variability in nigral expression, and what you're looking at in the blue is genetic variability in cortical expression that's driving the disease. Or it could be that what you're looking at in red is the genetic variability in resting expression, and what you're looking at in blue is genetic variability in expression in the context of amyloid deposition. It could be that amyloid deposition is driving this, and that's the key point. But the fundamental point is that these two diseases, which have similar histopathology but different neuroanatomy, have different. The evidence is that it's different genetic variability in the control of expression, which is generating that difference. So that's one element, I think, of selective vulnerability. I'm now going to talk more generally about how we're using syndromes with multiple genes to try and dissect this idea of selective vulnerability in a way which is using many genes for a disease. And the syndrome we started with was ataxia. And the reason we started with ataxia, well, two reasons we started with ataxia. The first is, if you go to OMIM, there are about 35 genes for ataxia. So we have a great source, 35 genes we can look at for ataxia. The second is that the anatomy of the cerebellum is very straightforward, at least to a geneticist. There's just Purkinje cells and granule cells. And then what we did is we looked at which genes among these 35 were co-regulated. So we took the genes and we looked to see which of them were co-regulated in the in the cerebellum, because the cerebellum obviously is the tissue where ataxia strikes. And what we found was two groups of genes. One we called the light yellow module. This is what the output of the computer program calls it. And all of these genes were involved in calcium homeostasis. And not only that, when we looked at these genes in the Allen Brain Atlas online, all of these genes were expressed in the Purkinje cells. The other, which the computer program called the TAN module, ataxin, here they are here, were all involved in the ubiquitin proteasome system. And when we looked at those genes were expressed only in the granule cells here. So we have this remarkable coincidence of events that genes involved in calcium homeostasis, when dysregulated, the cells which are most severely affected are the Purkinje cells, and those give ataxia. And then again in the ubiquitin proteasome system, those genes are in the granule cells. So how I see this is that for some reason to do with their function, Purkinje cells are particularly sensitive to the possibility of calcium dysregulation. And the most likely way for you to manifest a calcium dysregulation is as an ataxia. So we then, having taken all the genes for the ataxias, like I say, we decided to go through all the other disease areas. And when we did this for Parkinson's disease, we found that a very large proportion of the genes, only in the substantia nigra, showed evidence for co-regulation. And those genes were involved in mitochondrial complex 1. And interestingly, when we did this analysis, we identified this gene, RAB39B, as being important, as possibly important in Parkinson's disease, because it showed evidence for co-regulation with these other genes involved in complex 1. And then subsequently, this gene was identified as a cause of X-linked Parkinson's disease. So to some extent, our analysis predicted RAB39B's association with Parkinson's disease. And so this is saying that if you have a deficit in complex 1, then the most likely way for that to manifest is as Parkinsonism. And I should say that the pathologists, actually, people working on the pathology of Parkinson's disease, had actually said that they thought there was a deficit in mitochondrial complex 1 in the early 1990s. So this fits with much earlier work from the pathology of the disease. So this then is pointing at the problem in Parkinson's disease being in mitochondrial complex 1. So then we decided to go on to FTD and ALS, and this is the list of FTD and ALS genes. This list is current of about a year ago, but the list grows very quickly at the moment. And it groups this set of genes here, C9orf, VCP, P62, optineurin, and ubiquilin-2. These are genes which cause either frontotemporal dementia or motor neuron disease in different individuals, sometimes a mixture, but they can cause different syndromes in different individuals. And when we look at these genes which cause FTD and ALS, what we see is that these are all genes involved in the ubiquitin proteasome system. These are genes involved in the ubiquitin proteasome system, implicating autophagy as an issue for pyramidal neurons in the cortex in these diseases. When you look at the genes for frontotemporal dementia, many of them, not all of them, but many of the genes for frontotemporal dementia, granulin and CHIMP2B for sure, and then the gene identified in this, which was TMEM106B, this is also a lysosomal gene. Progranulin is a lysosomal gene. And when we did the GWAS for frontotemporal disease, we also came up with lysosomal genes. So the problem in simple frontotemporal dementia, in those dementia syndromes, at least a proportion of them are a problem in the lysosomal system. So I would say that the catastrophic cliff that pyramidal neurons are close to is a lysosomal collapse, basically. So my overall suggestion then, to finish, because I want a glass of white wine too, is that selective vulnerability is in part a consequence of different neurons having different weaknesses, being closer to different catastrophic cliffs based upon their functions. And the examples I've given you are Purkinje cells, calcium homeostasis, nigral neurons, mitochondrial complex 1, and cortical pyramidal neurons, a lysosomal function issue, and motor neurons too, either a lysosomal or a ubiquitin proteasome system. So I think we're beginning to be able to explain selective vulnerability in terms of the function of the genes by which cause these diseases. Thank you very much.